Our cells do magic every day, and every time they divide, they keep repeating this magic, right? So we will try to understand this a little bit better with something in our hands, like this tiny ball, and it is give or take about an inch wide, right? So I like you to, I don't know, you don't need to say it out loud, but I like you to think and take a guess at like how much smaller a cell, and especially the nucleus of a human cell, would be compared to this ball. We know it's tiny, but like how many times? It's like 1,000 times, million times, 100 times? If anyone wants to take a guess. Million times? Trillion, yeah. I think the trillion is correct. So two ways that we can look at it, right? This is a cell nucleus is about 6 microns. So this is about 6 micrometers, 6 in 1 million of a meter. When you think about it, it's 25,000 times smaller than the ball you have in your hand in terms of its diameter. But if you remember your math and then like how volume is computed, it's like 20 times 25,000 to the power 3. So it's like 15 trillion times, the ball you have is 15 trillion times bigger in volume compared to a cell's nucleus, right? So it is amazingly tiny entity, the most important entity in our cells, and it packs all this DNA. So second question. The string or the cord in your hands, it is about 6 feet, right? It's taller than I am. Anyone want to take a guess as to how long our DNA is when you stretch and add all our chromosomes on top of each other? How long of a DNA we would have in one single cell in our body? Well, this one is easy when you know it. It's the same size. It's the same exact size as the string that you have in your hand, right? So that's amazing. Now you're thinking and talking about something that's like 2 meters and fitting into a space that's like million times smaller in radius. And how does that happen, right? Our cells do that every day. And then if you like to try to put the string into that little ball, you will, when you're folding it, you will understand that this is not an easy task, right? So this string should fit. It's kind of amazing that not only it fits, but when our cells do it, they do it in a non-random manner. They do it in a very organized, very structured manner. So this is just a depiction of it, but we have this spherical nuclei, and then in it we have all of these chromosomes, and then each one of them fold in a certain way, and then each one of them do this without tangling within itself or tangling with the other ones, because they open up, they close back, they fold in specific patterns. When you divide the cells, they segregate again. So they do this in a way that is highly complex, non-random, and changes from one type of cell to another, right? I like to talk about the brains behind our research and talk a little bit about what computational biology is, what my journey is. But these are the people, the bright minds that we have that are making all of these happen, right? So you have my undergraduate students who are right out of high school, started working with us, like Lily and Kira in the front. We have Daniela and Laura coming all the way from Mexico. One is a Ph.D. student. One is waiting her Ph.D. offer letter these days. We have Abhijit, Nirmalia, Surya, Paramita. They came all the way from India after their PhDs or after their degrees there, and have been with the lab for quite some time. And me, myself, my journey, both I had a geographical journey and a little bit of a journey in terms of my scientific interests. So my journey starts in Turkey, where I was born, and I grew up there. I got my degrees in computer engineering and mathematics. So I had really nothing to do with biology. But then I came here and I did my PhD in a computer science department. There I started more and more being interested in how can we apply these algorithms and these methods that we develop with computer science to things like biology. And then after that, a little bit Boston and then Seattle. I did my postdoctoral fellowship in an institute, in a department that's kind of one of a kind. It's Genome Sciences department. So I was more shifting towards the genome studies. And I was in Northwestern University for a year. Then I came to Jolla Institute. And here I both established the lab and my family, and I've been here since 2016. So what is computational biology, right? That's in my title, that's how I describe myself. And then if you ask it to different people, different experts, they would have different opinions. One expert I asked was my kid. And then when I had to go for a seminar, I said, I need to go. And then he's like, Dad, why do you go? I'm like, I'm a scientist. I just need to go talk to other scientists. He said, like, Dad, you are not a scientist. You just do stuff on your computer. Right? I mean, in a way, it is honest and true. But kids these days are smart, but they're also brutal, right? And then he's right. He's right. This is exactly what we do. Like, I do stuff on my computer, but I do it in a fancier computer with multiple monitors, right? So at least he should give me that. But in terms of like a real expert, this is Bob Murphy, who was the chair of one of the most prominent computational biology departments, maybe one of the first ones in the country in Carnegie Mellon. This is his opinion as to how to describe it, right? So it is a science that tries to answer this question, how can we learn and use models of biological systems constructed from experimental measurements, right? So there's multiple parts to it. So the experimental measurements part, so we have many different ways of measuring our cells, our tissues, our health, right? So these can be levels of metabolites, these can be imaging certain cells and markers. This can be sequencing. And then we can do this in many, many different systems, biological systems. We can do it in human. We can do it in organs. We can do it in mouse. We can do it in other model organisms. And how we model all of that data can also vary. We can use statistics. We can use mathematical models. We can use machine learning, or more of the kind of thing that has had a huge hype these last year, the deep learning and then these large language models, right? Depending on what kind of complexity is needed. So, but everything starts with being able to, so this is the most important task, and I'm trying to also teach my students that. Most important thing is to find a problem that is worth solving, and then a biological problem or from our immune system or a biomedical problem, and then try to frame it or formulate it such that it can be solved with computational methods, right? Because this is the most difficult task, right? Once you formulate it, we know we're good computer scientists, we know how to solve problems that are formulated, but formulating it requires a huge teamwork, and that's why places like Jolla Institute for Immunology, these interdisciplinary institutes of research are critical, because to do that we need multiple different types of expertise, and we have that within one roof. All right, so another thing, I think computational biology, when people ask what's important in computational biology or bioinformatics, is that we need to be doing things that enable asking questions that haven't been asked before, or that enable capabilities to scientists that weren't there before, or enable discoveries in the respective fields that the scientists are interested in. And we do that through developing computational methods, and then they are applicable to from all the way to cancer, to infectious diseases, to autoimmune conditions, to development, and again studies of these model organisms, all kinds of things. But bio is important here, right? We call our field computational biology or bioinformatics, and both of them have bio in it, and they are front and center. So, so I like to talk to you about a little bit the genetics versus epigenetics, because when they ask, I'm a computational biologist, and what do you do? I say we work in epigenetics. So genetics is, you can think of it as all the ATCGs, right? What makes up your DNA. The sequence of your DNA that you can see on the right side, that is genetics. And that's something that is set there, and unless you have mutations or unfortunate things like cancer, it doesn't change much. Whereas epigenetics is this set of chemical modifications on DNA, as well as the how DNA is wrapped around these tiny complexes called nucleosomes, and then how they are then structured further and then fit like make a chromosome, and then how those chromosomes are fitting into our nucleus of our cells. So the way to define epigenetics is already hidden in the name. So epi means in Latin, epi means beyond. So epigenetics means beyond genetics, things that are beyond genetics, right? And our epigenetics is something where we have a little bit of control over through diet, through lifestyle choices. We are able to change like a little bit of that landscape of how the chemical modifications go, and so on and so forth. And then this, for instance, epigenetics may also explain why certain siblings have a disease and certain others not. Whereas genetics is the underlying cause. For instance, if you have an identical twin, sorry, it's the other way around, of course. Like if you have identical twin, your genetics is going to be 100% the same. So the only thing that changes is epigenetics, and that, as you know, has a huge impact. So when people say things like, oh, it's in your DNA or it's in your genes, that's generally used kind of like exchangeably, right? So we think that our DNA is genes, but less than 2% of our DNA is actually genes, right? So there's this vast amount of information in our DNA, more than 98%, that do not encode for genes. So, and that comes in play in many, many different places, but one place where it would help you contextualize something like the central dogma of the biology, which says DNA is a molecule that can replicate itself or that can encode RNA. And RNA is what gets translated into proteins, and proteins are what carry out the functions in our cells, right? So those are all true, but not complete, right? Because proteins go back and bind to DNA. And then when they bind, they generally bind to places that are these non-coding regions. All proteins go bind to RNA and help degrade them or keep them stable. RNA goes and binds back to DNA. So there's all kinds of these other complexities that we cannot explain only by the 2% of our genome. It requires the whole 100%. And this 98% has all sorts of biochemical activity that is different from one cell type to the other. Sometimes in diseases it differs. So that's important place to study. So both our genes and our non-coding DNA together determine a good portion of our disease risk and response, right? So we have seen this firsthand during the COVID pandemic, that we would be all infected with pretty much the same virus and then have a huge amount of, variable response from having nothing to unfortunately lots of deaths. So this is this is being determined by both the genes and mutations in the genes and changes in the genes, but also changes and differences in non-genic regions that act upon genes and then control how they are functioning. So after the Human Genome Project completed the very first full human genome in 2003, there was a huge hype and hope about this simple idea that now that we can make these maps, let's take any disease, we would be able to get DNA from the patients with that disease and then from the healthy people and simply compare. If we have enough numbers, then simply compare the two, and then we should have all our answers. What's happening in the patient DNA, in the disease DNA, that determines why this happened, right? So This had some successes and has given us a lot of information to work with, but it hasn't been to the degree that people have imagined. And then this brought us a new understanding of the complexity of how our genome works, and then triggered this whole era of epigenetics research that we are also now working on. So two issues of that GWAS genome-wide association studies, there were two issues. One, that it seemed to not give us everything we want. It seemed to give us less than how we can explain why diseases are heritable, right? How you're getting a disease and your sibling doesn't, and so on and so forth. So all of the DNA locus or DNA regions that people found associated with the disease, when you combined all of them, you would be still missing like how to explain heritability. For instance, for height, right? So there were hundreds or thousands of different regions that associated with the people's height, but yet when they are combined, they explain not even half of the variation of height. And in another way, it gave us too much too. So think of this Manhattan skyline, and then as you can see, there's a lot of buildings, and all of them I'm pretty sure are important, but some stand out, right? Some stand out, some are taller, some are likely more important than the others. So when we visualize the genome-wide association study data, we call them Manhattan plots because they look like Manhattan skyline, right? So you have a whole bunch of regions in the genome that are not necessarily associated with the DNA, with the given disease or trait, but you still have tens, sometimes hundreds of regions that are associated with the disease, but we don't know which one causes it, we don't know how they function, and so on and so forth. So in a way, like GWAS, genome-wide association, gave us too much and yet too little. That has made us scratching our heads as to how we can explain these disease associations. So one reason for it, and of course I'm gonna like go back and tie everything to chromatin folding, that's my job, but one reason for it is that when we have found regions in the DNA that are disease associated, such as this specific region where if you have a T, you are more likely to be healthy. If you have a C, you are more likely to have the disease that has been studied. When you look at it, it is not in the gene coding regions, right? So now, how does it function, right? Its function has to be through some genes, but we don't know which one. It can be through the gene Y over there because it's the closest one, or it can be through gene X, or it can be through a gene that's much further away in this linear space. The reason why we cannot know which gene in what cell type or what condition is the target of that specific region is that our DNA doesn't live in this one-dimensional space where we can say, okay, this region Y is closest, right? Our DNA, our chromatin, as I mentioned to you, lives in this three-dimensional space. And then this changes from one cell type to the other, right? It's not the same folding when you look at your retina versus when you look at your blood cells. It's quite different. So that makes life more difficult, right? How now you have all of these associations, how can you link them to what kind of target genes that they may be acting upon? So if we only had a way to measure distances, right? So you want to look at any given cell, and then you want to say, I see this region, and I see this gene right next to it, whereas this other gene is quite far away from it, right? So you can think of this, I think I like strings and whatnot, but you can think of this string as your nucleus or the DNA folded, right? So if I'm able to maybe get some reference points on this string, right? And then if there was a way to measure how far each pair of dots to one another are, maybe that I can use to reconstruct the whole structure, right? So that's in 3D. Let's start with 2D, and let's think about maps, right? We are thinking about genome maps and chromatin maps, but let's think about regular maps, United States map, particularly here. So here you are given the distances between each pair of cities, from Atlanta to Chicago and so on and so forth. 10 major cities across the two coasts and up north and south. So can you, or can we, by just using these distances, create the United States map, right? So we can. If you put these distances through an algorithm that does an optimization to try to find these two-dimensional coordinates of each of these cities such that those distances are best reflected, then you essentially get pretty much something very similar to the US map here, right? So this is the kind of thing you can do with computational modeling. So going back to our problem, we have two issues. This is a two-dimensional map. We really want a three-dimensional map. And the other more important issue is that this is cities and the distance between them is fairly easy to measure, whereas now we are talking about, remember that tiny cell, much tinier than the ball you have. Now we are talking about the nucleus, a very tiny space, and measuring distances between pairs of regions in that tiny space, right? So how can we do that? In 2009, researchers in Massachusetts, Boston and UMass, they came up with this very revolutionary method where they wanted to look at how DNA is folded in a given cell, and they said, maybe to be able to do that, we can use some biochemistry and molecular biology trickery in a way that, like, we take a snapshot of it, we keep it stable so that we can do further experimental steps on that and measure it. So on the left you have a nucleus and how DNA is folded in that, and then when you use something like formaldehyde, a cross-linking agent that keeps regions that are close to each other intact. So that's those little cuffs over there. So while you keep them intact, then you can introduce things called restriction enzymes. These are actually immune defense for bacteria. They chop off virus DNA with these molecular scissors, but we can produce and use them. So you cut the DNA so that you have smaller pieces to deal with. And then while they are being held together still, while these proximities are kept, you can religate them back. So now you have this circular piece that holds that information about who was close to what, right? And then you chop it one more time now, and then it's linear, it becomes linear, and it tells you maybe this yellow region and this red region, I found them close to each other in one cell. The reason why we want to do it linearly is that sequencing likes linear DNA, right? So then we do a sequencing on this, and then these are very expensive experiments. So we do literally billions of reads, and this is tens of thousands of dollars, maybe more like $5,000-6,000 these days with the sequencing costs going down. But it gives you this map that we call contact map. So how much each region contact another. So this is very similar to, or from this you can really easily get a distance map, right? So this is how proximal they are. You can convert this, and then you can look at how what's the distance between each pair of regions. And as you remember from those kind of distance maps, we were able to do like a 2D map creation. We can also do a 3D map creation. That part I'm not going to go into details, but at the end of the day, if you say nucleus is this big and here's my distances and avoid collisions and tangling, you're able to get a 3D model of our chromatin. So these studies have been revolutionary in our understanding of the 3D genome organization of our cells and of our chromatin. So multiple layers of information it brought. Some of them were a little bit already known, like such as this squares that you are seeing. This is all chromosomes by all chromosomes. We already knew that chromosomes fold first within themselves and then kind of like have this interface with the others. And then this one was also somewhat known from imaging studies, that you have these two broad chunks of chromatin. One is this active regions where genes are expressed and there's active biochemical marks, and one is the inactive regions. And then what these maps showed us, this checkerboard pattern, told us that the active ones like to talk to active ones and inactive ones like to talk to inactive ones more. But of course, the high-C or these type of experiments made it possible to do it in many, many cell types at a higher resolution. So the kind of things that completely were unknown and then were enabled by these type of methods is what you see in the right-hand side when you zoom in one more time. These square blocks on the diagonal that started emerging, which we call them domains of interaction. And then if you can see those tiny dots at the corners of those domains, which correspond to, in the figure, if you see that two anchors marked with blue dots. So those are the anchors that keep that region intact and interacting within, but not across. So we call them loops, right? So what do these loops do, and then how do they occur? How are they formed? So I wanted to have something cute because the real mice figures may not have been. Here you are looking at development of mouse embryo, right? And we know a gene called Sonic Hedgehog, named after the Sonic the Hedgehog actually, that is very important in multiple steps in development and also in this limb development, the development of legs and arms. And then as you can see, the activity of this like distal region called ZRS enhancer is happening at the specific developmental stage at the limb regions, right? And the fascinating thing is that enhancer is really nowhere near this gene. It's 1 million base pairs away, right? And there's like 10 or so other genes in between that enhancer and then the Sonic Hedgehog gene. But through mouse genetics, this enhancer was found to be controlling the gene, and then through these looping studies, then later it was confirmed that how it controls the gene is through looping, right? So when you take away that enhancer, and then remember, yes, sorry for the little guy, but remember that the enhancer has nothing to do with gene mutations or gene coding region, right? It's just a non-coding. It's one of that 98% that doesn't code for genes. When you take away that enhancer, mouse end up not having limbs. Limb development has not occurred, right? So you can say that this is cool and cute, but why do I care about mice, or why do I much less like care about a cartoon mice? But same things happen. Same enhancer is there in human, and it controls also the limb development. And then in the mouse case, it was a whole deletion of that enhancer. In the human, it is essentially these natural genetic mutations that are appearing, just changes of one base pair from a C to a G, for instance, leads to these developmental abnormalities such as polydactyly, having more than 5 fingers, right? So this is not just a one-off case, right? A lot of genes have their control switches or enhancers much closer to them than this 1 megabase, but this is by no means a one-off case, and there have been cases where after these looping studies, people have kind of like threw away like 10, 15 years of literature, and they have been looking at the wrong gene all along because that gene had nothing to do with that disease. The enhancer near that gene was actually regulating something that's very, very far away. So you can, another way of contextualizing loops is, or enhancers with loops is that you have these control switches that give the genes the green light to go and express and function, right? And then when the gene is proximal to that control switch, the activity is on and happening. And then when they fall apart, when they don't see each other or loop to each other, that activity is severely diminished. So how these loops are formed, we have a nice animation thanks to a lab in MIT. I will show that, and we'll try to also see if we can make you listen to the nice music. But this animation essentially will show you how you can go from on the left side, this boring linear structure of the DNA, to the one on the all the way to the right, where it's looped and created these domains. So let's try. So you are seeing the chromatin here, or the DNA, together with two other players. One is CTCF. It's those red ones that act as insulators. And the other one is cohesin that falls onto DNA, extrudes the DNA until it sees certain orientation and certain types of CTCF and stops, and it gets stuck. So that is exactly how one loop is created right now, as you can see. But the thing is, all of these are dynamic. Those CTCF stay on the DNA for only a certain amount of time. So does the cohesin, right? They fall off, but then they get reloaded, right? And then now they get reloaded into different places, and cohesin still extrudes. Sometimes they collide into each other, right? So that's how exactly you are seeing that two-domain structure that cohesin creates. And I'll just have you enjoy the rest of it. I mean, the music is nice. Thank you. Thank you. I know that's for the music. One last bit there that was important was that where it was saying these contact patterns that you are seeing in the little heat maps in the bottom, they emerge from many cells aggregated together, right? So all of these cells doing this waltz, and then when we do these experiments, we use hundreds of thousands of such cells, and then in aggregate, these patterns emerge, and then they give us a good idea as to what is close to what in a given cell nucleus. How can we find those loops, right? So I like showing nice, beautiful pictures. I don't know if it has something to do with the loops, but for me it has. So you see this sunflower field, and then again, if you're reading about AI and so on and so forth, you could imagine that one can try to write an algorithm, write a computational method that detects sunflowers on this picture, right? So the most important thing that they need to, I mean, there's a lot of things that they need to factor, but the most important thing is that the sunflowers appear in multiple scales, right? I mean, finding the one in the front might be not too difficult, but how about finding the ones that are all the way far away? So they come in different shapes and sizes, right? This is also the same thing for loops. They come in different shapes and sizes. So a postdoc in my lab who came from computer vision field or image processing field have taken this idea and this formalism called scale space theory and then applied it to these contact maps. I'm not going to go into details, but by like iterative smoothing of these contact maps that you are seeing together with other filterings, we were able to find in a very efficient way all sorts of loops of all shapes and sizes from these contact maps. And this algorithm has been very popular and applied to many different problems, from again mouse brain development to like immune cell gene regulation and model organisms. So our lab does a lot of different projects, but I just want to highlight a couple where I can do with more pictures. So one of them is this project where we have started with the idea that these three-dimensional maps, they will also help us because like when genomes are rearranged in cancer, which is what happens, the places get deleted, inverted, or translocated into other chromosomes. So these contact maps immediately tell you where this is happening, right? So we kind of like developed algorithms that solve those, but then we looked for more complex rearrangements, and then we came across this one called chromathripsis. So this is a dramatic representation of it, but it's pretty accurate too, that in chromathripsis, it's a catastrophic event. And unlike these cancer mutations that acquire like over time, this is like one-time event that breaks almost the whole entire chromosome into pieces. And those pieces get shattered and they actually get shuffled. Some of them get lost, some of them get amplified, but then they get put back together. But now the whole chromosome ordering is messed up. And then you can imagine how this can lead to like genes breaking into into two, or two parts of different genes coming together and fusing, creating what we call fusion oncogenes. Or if you have, you know, like already become aficionado of the 3D chromatin, you can imagine that these rearrangements can bring those enhancer elements or control switches next to genes that are not supposed to be active, right? It might be an oncogene that hijacks such a switch and then will be activated at high levels, and then those cells will have some sort of growth advantage. So when we looked at the chromathriptic chromosome, the heat maps on the left looked like a mess. I mean, they are far from those ones that I showed you, like the nice diagonal, and then it kind of fades away. But we were able to develop an algorithm that finds all of these breakpoints and then essentially like Rubik's Cube, shuffles them back in place and then finds the corrected ordering of a chromosome from the patient's DNA, right? And then this was, in our case, this was done with a rare and aggressive subtype of B-cell leukemia in pediatric patients, where if they have this mutation or if they have this chromathripsis event, in the past, in like 15 years ago, they were like 5 times more likely to die. And then after this has been figured out, they are now being treated at much higher dose, but they are still at a higher risk of relapse and a little bit higher risk of also, unfortunately, death. So this is, you know, putting the chromosome and the correct ordering back together for a given person's DNA allows you to look at specific events, specific mutations that may be driving their cancer. I can talk about more projects in the lab, and you know, I'll stay here and then answer as many questions as you like. But I think I want to also answer the question maybe that's in your minds, that like how will any of these help me, if at all, right? Or what day, when this will happen? Why are you doing this research, right? So it does actually help already people of certain type of diseases. It's groundbreaking times. It's very exciting times. One disease that has been now the pioneer in the like cellular and gene therapies has been what's called sickle cell. So this is a disease about red blood cells. They carry oxygen around. And then in adults, we have hemoglobin called human adult hemoglobin. And the mutations in the adult hemoglobin kind of mess up with the cell's function and also structure. And the structure is also important because they go clog the arteries, and it's very painful. For those who are African descent, this disease is even much more prominent than any other ethnicity. So it is a huge health burden. There's 300,000 people born every year with this disease, right? So just recently, end of last year, it was announced that FDA approved the very first gene editing therapy for sickle cell disease. And this was followed more recently past month on like the approval of the same for beta thalassemia, which is also a hemoglobin disease. And this was mainly, you might have seen this, and then this was mainly, of course, touted as a success of CRISPR-Cas9, this gene editing technology. But although that is the technology that enabled it, I'll try to tell you a little bit that it was the type of research that I talked to you about was the main reason of the discovery that gave you the target to be edited so that this becomes a cure. So back in 2021, this first paper came, and then they have tried it, and they have shown that what they were able to do is now that the adult hemoglobin is mutated and it's difficult to fix those mutations, they were able to activate fetal hemoglobin, which does also the same thing, but just does it before birth and kind of switches off after birth. The reason, or like how it all started to get to the fetal hemoglobin being like the cure or being like the alternative mechanism to the curing this disease has been one of those Manhattan plots, right? One of those genome-wide association studies where in 2008, this is Stuart Orkin and others from Boston Children's, they have conducted such studies, and then they have found regions that are associated with the sickle cell. And then as you can see that HBB, HBA, those are hemoglobin genes, and they are already not a surprise. We already knew by then, I think these are mutations in the hemoglobin genes. But what is surprising is that BCL11A, so it's a gene on a different chromosome and has nothing to do, at least to the best we knew at that time, with hemoglobin. But then further studies by the same group and others have shown that BCL11A is actually a protein that controls the expression of hemoglobin. So when you have high levels of BCL11A, which adults have, the expression of fetal hemoglobin goes down. So it suppresses fetal hemoglobin so that we switch to adult hemoglobin. And it does it through this interesting looping mechanism, right? So it's all about enhancers and loops here. And then in addition, they were later able to map. So you really need the hemoglobin to be working in erythroid cells and not other cells, right? Through these enhancer studies, these epigenetic studies, later the scientists were able to map a specific enhancer that's only controlling this gene's expression in erythroid cells, in red blood cells. And that is what enabled actually this cure. The Casgevy actually doesn't correct the hemoglobin mutations. It deletes that enhancer. And if you remember, that enhancer controls this gene that suppresses fetal hemoglobin levels. So once that enhancer is gone, BCL11A levels go down, and fetal hemoglobin is free to be expressed again. And now you have a functioning hemoglobin, not the adult version, but the fetal version, which is quite effective. And these patients have, after 18-month follow-up, they have been 29 out of 30 were cured, and then they didn't need further therapy, such as the hematopoietic cell transplant, and that kind of like gives you an idea as to how this is already impacting our life. So I want to give you some more time, and I'm running out of time, so I'll stop here. I mean, this is a really nice science art collaboration that we had a couple years ago at the San Diego Art Institute, and we worked with a scientist, and I told her about all the things that we are fascinated about, and then she made these really nice, beautiful art pieces. With that, I will stop. I will thank, of course, all my lab. We love ice cream, we love food, as you have seen in the other picture. And then all my collaborators, long-standing collaborators, funding agencies, all the LGI communications team, Matt recording me here, Sonia, Jenna here, and then Gina also for feedback on the presentation. And you know, we are a very international lab, and that really helps us have interesting ideas. So And thank you so much for coming here and braving the weather.